have all recovered from last week's failure, and that you could squeeze in a little bit of time between fieldwork to join us today for this very exciting, unfortunately, online only talk. But we're still nonetheless really happy to have Andrew Mehring joining us, who's joining us from the University of Louisville in Kentucky. So he's an assistant professor there, working on ecosystem and ecosystem function work. So many of you will know Andrew because not so long ago he was a postdoc at the biological station of Doñana. And I'm really excited because I think it's been a while since we've had a talk focused specifically on a project carried out in the national park. So before I hand over the word to Andrew, just a very quick introduction. He did his thesis, his PhD in Georgia on the East Coast of the United States. He then moved around a lot. So since then he's been in California and San Diego, and that led him to also spend some time in Australia and Melbourne before coming to Spain, to the biological station of Doñana with a Marie Curie postdoc fellowship with a secondment in the Netherlands. So about a year ago or so, he left us for Louisville, but we're really happy to have him talk about his work. So take it away, Andrew.
Okay, thanks very much for that introduction, Chris. And it's really great to be able to present at the Doñana Biological Station. I'm really sorry I couldn't be there in person. I miss Sevilla very much, although I'm very happy to be in Louisville, Kentucky now. Anyway, today I'll be talking about zoogeochemistry and the effects of flamingos and invertebrates on wetland greenhouse gas emissions. And this I do want to acknowledge again, as Chris said, this work was funded by a Marie Curie individual fellowship. So I am an ecosystem ecologist, as Chris mentioned, and I know we don't necessarily have a lot of people at EBD who consider themselves ecosystem ecologists. So I'll just briefly talk about, what is ecosystem ecology. I had a friend once who is a disease ecologist ask me, so an ecosystem ecologist, does that mean that you're like an ecologist? But actually what ecosystem ecology means is that we take an ecosystem, and a lot of times the boundaries of the ecosystem are defined by the researcher, and we measure the flow of material and energy into and out of the ecosystem, and also the cycling of material and energy within the ecosystem. And so I can summarize the whole field with this very simple diagram that you see on the screen right now. Personally, I spend a lot of time, I mostly started out working in aquatic ecosystems, wetlands, lakes, streams, and rivers. And I also conduct a bit of research now in forest, terrestrial ecosystems, and also in urban ecosystems. I'm specifically interested in biotic effects on ecosystem function. And by biotic effects, I mean the effects of plants and animals on ecosystem function in both aquatic and terrestrial systems. And specifically, I spend a lot of time now studying greenhouse gas emissions and carbon flux, especially carbon dioxide and methane. And I'll talk a little bit about why I'm interested in measuring these things. When I talk about flux, I'm actually talking about the movement of matter from one place to another. So why do I want to measure carbon flux? Well, we can learn a lot of things by looking at the movement of carbon atoms through ecosystems. For example, this is some research I was conducting in Southern California. We can take a box and put it over a plant if it's sealed. And by measuring the decrease in concentration of carbon dioxide atoms inside of this box, we can actually measure the rate at which plants are removing carbon dioxide from the atmosphere. Or another example from Laguna de Santa Olaya during a very dry period when the laguna is very green. We can take a floating chamber, put it onto the surface of the wetland, and then I can actually measure increases in carbon dioxide or methane concentrations within this chamber, and then I can calculate the rate at which those elements or molecules are leaving the wetland and escaping to the atmosphere. And what I'm actually working my way into now by placing open path sensors above an ecosystem mounted on a tripod or a tower, we can measure the emissions of carbon dioxide or methane from larger spaces, from whole ecosystems, without the effects of these chambers, for example. Now, why do I love ecosystem ecology so much? Well, just an example and how it relates directly to us. Humans store carbon. You began basically as a cluster of cells, and certainly by the time you were 6 months or a year old, your body contained more carbon than it did, at the time of conception. And of course, now your body contains much more carbon than it did when you were 6 months or a year old. So we basically store carbon. You are, as a human, if you look over here, about 18% carbon. So you can take, if you want to figure out how much carbon your body is storing right now, you can take your weight and multiply it by 0.18, or if you want to be really simple, just divide by 5. And more or less, that'll give you a rough idea of how many kilograms of carbon are in your body. Now, where did all that carbon come from? Well, literally, the carbon that is in your body came from the food that you eat. And I think we take that for granted a lot, but actually the atoms of carbon that are in your body were earlier, maybe yesterday or a few days ago, the carbon that was contained in this salad, the carbon that was in this burger or pizza or this fish. And then you actually took those carbon atoms and stored them in your body as carbon. And those carbon atoms, more or less, have always been carbon atoms for hundreds, thousands, even millions of years. So if you think back, the carbon atoms in your body potentially were a part of many other things. So at some point, whether you're eating, you're a vegetarian or if you eat meat, at some point that carbon was cycling through a food web. And at one point that carbon, that food web was probably relying on plants. And so those plants actually got their carbon from other organisms that had lived before them. For example, you have this insect, this katydid, which is what we call them in the United States. It's an orthopteran. And say this katydid was feeding on leaves in a tree, and then one day the katydid dies, and it begins to decompose. As the microorganisms break down the tissues of this insect, carbon dioxide is released, and that then can be taken up by the leaves of the plant. So the plant now contains carbon that was once part of a katydid. And the plant is, basically, sharing elements or has taken elements from this other organism that lived before it. So you could also think that there's a good probability that some of the carbon atoms in your body right now were once part of the carbon that was contained in an insect. You could think even farther back, and there's a very good probability that some of the carbon in your body right now was at one time part of a dinosaur. And there's an even greater probability, if you think even farther back, in fact, a near 100% certainty that the carbon in your body was at one time part of a dying star. And so these are some of the ideas of ecosystem ecology, that basically elements are constantly moving between the inorganic components in the environment and also between organic components of the environment, and are basically passing from one organism to another. Now, I don't mean to get too heavy, but at some point you will give back this carbon. Of course, your bones will remain for a long period of time and actually will store carbon for a long period of time as long as they're intact. But you will release some of that carbon back to the environment. And so the carbon that is now in you, maybe at some point will become a part of a coral reef. Maybe it'll become part of a fish or a turtle that you see here, might get incorporated into a huge tree and stored for long periods of time in the wood in that tree. Or potentially, and very likely, could become part of another human. And these carbon atoms we're just borrowing them temporarily, and they will continue to cycle. But you don't have to wait until you pass away. You're actually doing this right now. When you exhale, when you breathe out, the carbon dioxide in your breath is not just something that was in your blood. You're also exhaling or breathing out carbon that was at one time parts of the tissues in your body that are constantly being replaced. And so there are little bits of you right now that are going back into the environment and being taken up by other things. And if you have plants in your house, there's a very good chance that those plants contain carbon that was once in your body. There's a great probability that those plants now have carbon that was once a part of you. And if you talk to your plants, there's an even better probability that those plants contain carbon that was once a part of your body. So I talked a lot about how humans store carbon, and that's not, of course, really describing our role in the environment. As you all know, we have a lot of problems now because humans take carbon that is deep underground, stored safely and sandwiched within layers of rock. And then, of course, we burn that carbon. And when we do, we're emitting massive amounts of carbon into the atmosphere which of course has been raising the concentrations of carbon dioxide in the atmosphere and contributing to climate change and warming and a lot of the issues that we're facing now, particularly in places like southern Spain, but all over the world. So this describes our role in the ecosystem, not just things that are cycling carbon, but also the way that we affect the atmosphere, the way we affect ecosystems and climate. Humans alter carbon cycling, and because we are animals, you could think about this as one aspect of zoogeochemistry. So zoogeochemistry, what is it exactly? I remember learning about biogeochemistry when I was a master's student. One of the other students, her partner was visiting, and he introduced himself and he said, "I'm a biogeochemist." And I thought that sounds awful, because it's taking three complex subjects, biology, geology, and chemistry, and combining them into one. Since then, I've actually come to love this. And if I was going to describe myself as something other than an ecosystem ecologist, I might say I was a biogeochemist. But biogeochemistry concerns the physical, chemical, biological, and geological processes that govern ecosystem structure and function. And ecosystem structure, of course, talking about community structure, and function being things like carbon emissions or carbon storage, for example. Now, zoogeochemistry specifically focuses on the roles of animals in biogeochemistry. Oh, and I was going to mention, if you were interested in reading more about this, there's an excellent review that was published in 2018 by Schmitz et al. In the journal Science. So a couple of quick examples that might be familiar. For example, we have a picture here showing in the Pacific Northwest of the United States, we have some salmon that are swimming upstream. These are diadromous fish. They spend large parts of their life, they basically are hatched in streams as from eggs. They spend maybe just a few years in the freshwater environment, and then they move to the ocean, and they spend the majority of their life actually living in the ocean as predatory fish. And then they return to streams when it's time to spawn, going back to their spawning grounds. They reproduce, and then they die. And those carcasses of the salmon are then, of course, they're fed upon by bears and wolves. They are dragged up into the forest where they decompose. And so basically these fish, animals like these fish, are really involved in the transport of elements all over the world. So these salmon are bringing lots of carbon and nitrogen from the ocean and bringing it back onto land during this annual migration of these diadromous fish. Some researchers, actually, this is some work that was published in the Canadian Journal of Forest Research. They're actually looking at salmon, these streams where there are large salmon runs, large numbers of salmon returning to the freshwater ecosystem, and comparing it to streams where there weren't many salmon coming back. And you can see shown in red on this plot on the bottom, we see this enriched signature of this oceanic or marine nitrogen being brought back to land. And they were taking cores of these trees and actually measuring the nitrogen isotopes within the tree. And they could see the signature of this oceanic or marine nitrogen in the trees and these streams that had large numbers of salmon returning. And as I mentioned, these carcasses of the salmon are dragged up into the forest. They're fed upon by scavengers. There are also decomposer invertebrates and other animals that feed on these carcasses. And what happened then, as a result, there are predatory beetles that the researchers captured, and also were able to measure that signature, that oceanic nitrogen within the beetles. And not just in the beetles, but also within the vegetation, like these salmon berries and huckleberries and things that are growing within the riparian zone around these streams. So salmon are moving massive amounts of nitrogen back from the ocean to land. Another example, some of you have probably heard about beavers. Beavers create dams and rivers and streams, which slows down the flow of water and creates these large wetlands. In doing so, all of the material that's flowing downstream will then slow down and drop out of suspension. And rather than continuing to flow downstream, it accumulates in the wetland. As a result, this is some research by Ellen Wohl and others that was published in Nature Communications. You can see we're looking at megagrams of carbon that are stored in these different types of ecosystems. And in the long abandoned beaver meadow, notice this brown bar down here is showing that there are over 3,000 megagrams of carbon per hectare stored in these long abandoned beaver meadows. And it's actually thought that when trappers reduce the number of beavers from some 500 million before European settlers arrived in North America to the lower numbers we have today, we actually potentially release lots of carbon into the atmosphere because of reducing the populations of those animals. So animals are very important for carbon storage. And also additionally, some work that I have been engaged in, animals also can affect the movement or the emissions of carbon as well. So these are leafcutter ants. I'm not sure how many of you have spent much time learning about these, but these ants create these massive colonies underground, and they bring in leaf material from the surrounding forest to feed a fungus that's growing within the nest. At some point, so that leaf material starts to become less useful to the fungus. So the ants actually transport the used leaf material to an underground refuse dump, like a landfill, a place where they dispose of their trash, and it accumulates there. And if you've ever seen pictures of how large these nests actually are, these are some researchers who poured concrete into a nest and then slowly, painstakingly dug away the dirt around it to expose this massive leafcutter ant nest underground. We were looking at these nests while we were working in Costa Rica, thinking that, it's basically these ants have these large underground trash dumps or landfills. And as our human landfills emit carbon dioxide and methane to the atmosphere, there's a good chance that the leafcutter ant nest is also acting as a landfill and emitting carbon dioxide and methane to the atmosphere. And we measured those emissions, and you can see this is methane flux in milligrams of carbon per square meter per day on the y-axis. And you compare the non-nest soils to the soils where we have nests, and you can see that those emissions of methane can be greatly increased from leafcutter ant nests. So, and then this brings me to some of what we're specifically interested in the project I'm talking about today. Now, benthic invertebrates, as an example, these are invertebrates that live on the bottom of wetlands, enhance greenhouse gas fluxes. This has been shown in a lot of different, primarily laboratory studies, that through a number of different ways, invertebrates like aquatic worms, these fly larvae, this midge that you see here, can enhance the flux of nitrous oxide, carbon dioxide, and methane, our three most important greenhouse gases. They can enhance the movement or the release of these gases from sediments in aquatic environments. And they do that in a number of different ways. Worms, for example, are considered conveyor belt feeders. They feed head down in the sediments, feeding on organic particles. And then as they're digesting the particles, they move up through the body of the worm. The particles drop back down. And in doing so, this creates this constant cycling of sediment, organic matter particles from the upper layers of the sediment to the bottom layers. In these deeper layers of sediment in a wetland, there's usually very, very little oxygen because all the bacteria and fungi living in those sediments rapidly consume all the available oxygen. And so down in the deeper layers of sediment, conditions are basically anoxic, lack of oxygen. And it is in anoxic conditions that many of our greenhouse gases, such as methane and nitrous oxide, are produced. So worms might actually increase the rate at which particles move to these deeper layers, and this organic matter is exposed to anoxic conditions, and therefore more methane might be produced as a result. They also create tunnels that allow gases to escape more easily to the upper water and eventually to the atmosphere. And also things like chironomids will create U-shaped burrows, and they undulate their bodies to pull in fresh supply of oxygen. And also they pull in food particles and inadvertently pull in things like nitrogen-containing compounds like nitrate. And then in the burrow of the worm, because of all these chemical changes happening, you can see then increased emissions of nitrous oxide as the nitrate is broken down and denitrified. We actually might see both nitrogen gas and nitrous oxide being produced at higher rates. So a number of different complex mechanisms that actually have been shown to allow invertebrates to enhance greenhouse gas emissions. Now, several researchers at the Doñana Biological Station have shown that water birds can reduce the abundance of these very same groups of aquatic invertebrates. For example, things like snails, polychaete worms, and these fly larvae, this red aquatic midge larva. It's been shown that water birds like flamingos can actually cause them to have lower populations in aquatic environments. They also reduce plant biomass. This is an image that I think many of you may have seen before, and I should note that some of the images I'll be sharing here today are actually provided by many of the photographers and researchers at the Doñana Biological Station. I did not take this photo, but this is a photo of one of the wetlands in the Doñana natural space. And this is basically a plane flying over, conducting a water bird census, a count of the water birds in the wetland. And just before the plane flew over, this wetland was full of flamingos. What you can see here, these circular shapes, of course, are left because of the feeding activity of the flamingos. You're actually seeing their disturbance in the sediments. And what causes this is actually a video that was made in Laguna de Santa Olaya by one of my interns. Flamingos are feeding by doing this sort of a dance in a circular motion where they're stirring up sediments, and they're getting a mouthful of water and sediment and hopefully invertebrates. And then they're using these structures within their bills or within their beaks called lamellae, which act a lot like the baleen of a whale. So the flamingo gets a mouthful of sediment and water and invertebrates. It closes its bill, and then it has these structures that act almost like a mesh or a sieve, and they can push the water and sediments out through those spaces in between the lamellae, but they're perfectly spaced and perfectly sized to retain the invertebrates. And so they're really efficient filter feeders then, in a way, these flamingos. By doing this, they disturb the sediments. They might also reduce plant biomass, and they will also lower the numbers of invertebrates living in the wetland. So this was again, as I mentioned, a Marie Curie project, and the title of our project, the acronym, was Flamingos. And what that actually stands for is the acronym that you see there is functional links in avian, macrophyte, microbial, and invertebrate greenhouse gas output simulation. And I think that the acronym actually helped us to get this project funded. But we are specifically interested in these wetlands in the roles of invertebrates in greenhouse gas emissions from the wetlands, but also the roles of water birds in modifying these greenhouse gas emissions. So what is the general idea here? Well, we're interested specifically in the predatory effects of certain birds like flamingos on invertebrates and greenhouse gas emissions. So the idea is, if we have more birds, more flamingos, which is actually something that has been happening, we've seen that the numbers of flamingos have been increasing in a lot of these coastal Mediterranean wetlands over time. Also, numbers of ibis have been increasing as well. If we have more birds, this is possible then that we will have fewer invertebrates as a result, because these birds are actively feeding and keeping these invertebrate populations in check. And if we have fewer of the invertebrates that have been shown to be able to enhance greenhouse gas flux, then perhaps we will have reduced microbial activity of the microbes that would produce these greenhouse gases, and also have fewer emissions of methane, nitrous oxide, and carbon dioxide. And that is what we're trying to test. There are two different components of flamingos that I'll talk about today very briefly. One, we conducted a laboratory study, and this was at Wageningen University as part of the secondment. I spent 3 months there working on this laboratory study to measure the effects of invertebrates and sinking algae on greenhouse gas emissions. And then the second part, probably the part that was really the bread and butter of the Flamingos project, is we're looking at top-down effects of water birds on greenhouse gas emissions with a manipulative field study. So the laboratory study, you can see here, we were working at Wageningen University, primarily with Jeroen de Klein. And this is actually my partner, Meredith. She was teaching English in Spain while I was there on the Marie Curie Fellowship. And during the summer, she didn't have to teach, so she was able to join me there in the Netherlands. And we had these laboratory mesocosms that were being incubated at a controlled temperature in these water baths. They have really great facilities there at the Wageningen University. You can see these are containers that contain sediments from a local wetland that was sieved to remove all the invertebrates and control the organic matter content. And then we actually added to those mesocosms these aquatic worms. These are oligochaetes. This is a tubophysid. It's actually tubifex. We were able to buy it from a local pet shop, and because people use them for feeding their fish and things like that. And we actually, to these mesocosms, we could add worms at different densities, either no worms present, about 15,000 worms per square meter, or 30,000 worms per square meter. And I should point out that these densities are not exceptionally high. In fact, I've worked in other wetlands before, in some of the Australian wetlands where I was working in the past, we saw densities of these aquatic worms up to 500,000 per square meter. So these can be present in very, very large numbers. These worms are usually present in more, you see them in higher numbers, and they really dominate communities in wetlands that are more impaired oftentimes. So as water quality decreases from eutrophication, for example, if we were adding excess nutrients to the wetland and water quality is impacted, you might see more aquatic worms in the sediments. So then we also crossed this in a fully factorial design. We crossed our different densities of worms with different amounts of dead green algae. And we actually bought chlorella, which is a green alga. We bought this freeze-dried chlorella from de Notenschap, which I think means the nut shop in Dutch, because this is actually something that you can add to your milkshake or your smoothie if you're into health food and you want to eat algae. But we were adding different quantities of dead green algae because we were trying to mimic the effects of what happens in a wetland when it receives too many nutrients. When there's too much nitrogen and phosphorus entering a wetland, of course, that benefits the algae. And then you get these large algal blooms, which this is a process called eutrophication. And when you have these large densities of algae, of course, they begin to die in large numbers, and that can reduce the oxygen concentrations in the wetland, which can be very harmful to lots of other aquatic organisms living there. Because once the oxygen is reduced, many other organisms in the wetland cannot survive. As you add extra nutrients, of course, you're going to have greater and greater quantities of algae sinking from the water column down to the sediments below. So we're mimicking oligotrophic, mesotrophic, and eutrophic conditions, which are different levels of nutrient loading on the wetlands. And we had these algae being added at basically 7, 25, or 63 grams of carbon per square meter per year to mimic, different levels of being impacted by nutrient loading. And this is based on literature values of rates of sinking algae in these wetlands. And half of all these mesocosms were aerated with bubblers, which you can see over here. Our hypotheses were that methane flux would be highest at the highest levels of worms and of our algae, and that the effects would be dependent on oxygen levels. And you can see these are the results from the final week of the study. We have methane flux, this is the movement of methane, in grams of carbon, of methane carbon per square meter per hour. On the left, we have mesocosms that were not aerated, and you can see our increasing tube effects density. These are worms per square meter on the x-axis. And the different color codes, red would be that at low levels of algae, green would be medium levels of algae, and blue are high levels of algae. And you can see that as worm density increases, we see increasing levels of methane emissions, but this is highly dependent on the amount of algae present in the system. So there's this significant interaction of worms by algae by aeration. The effect of the aeration can be seen here on the right. Notice that when the mesocosms were aerated with bubblers, they had higher oxygen concentrations, and we can see that there is no effect of worms and really very little effect on the increasing levels of algae on methane emissions. So these effects are really dependent on the oxygen concentration in the system. So getting back to our original hypotheses, methane flux was highest at the highest levels of worms and algae, or chlorella, as you can see here in blue. But the methane fluxes were much lower in oxygenated mesocosms, as you can see here on the right. Similarly, for carbon dioxide, you can see we're looking now at carbon dioxide flux, and these are grams of CO2 per square meter per hour. And these are, again, same color code, low algae, medium levels of algae, and high levels of algae in blue. And we have tube effects density here on the x-axis from 0 to 30,000 worms per square meter. And notice that there is a significant interaction between worms and algae again, but this seems to be primarily driven by algae in this case. Notice that when algae levels are high, we're seeing high levels of CO2 flux from the mesocosms, regardless of the worm density. But at lower levels of algae, we can see there is this interaction where the higher the densities of worms are, the greater the release of CO2 from these mesocosms is. Now, the difference here, of course, we have similarly the carbon dioxide flux was highest at the highest levels of worms and chlorella, but carbon dioxide flux was not significantly affected by aeration. So in conclusion for this first section, invertebrate enhancement of greenhouse gas flux was influenced by the availability of labile organic matter, the sinking algae in other words. And the interactive effects on methane flux might be limited in oxygenated waters. So moving on to the second component of the big component of the Flamingos project, our manipulative field study. How did we manipulate densities of flamingos? Well, I've talked to other people about my dream of one day being able to fence off an entire wetland to keep all the birds out. I know it's basically, it's a pipe dream, right? Not likely to happen, but we've actually used techniques that people like Andy Green, Marta Sanchez, other people at the Doñana Biological Station have used before, where we use these exposures to keep flamingos out of a certain section of wetland and then measure the effects happening within the exposure. So these are basically constructed with 4 iron poles, iron posts that you can see here. And they're about 3 meters apart on each side. These, of course, are not drawn to scale. If they were, this flamingo would be terrifyingly large. But this is basically a distance of 3 meters on each side. And then a stainless steel cable is stretched around these 4 posts, and it's at about 70 centimeters off the bottom of the wetland. And that has been shown to be effective at excluding flamingos from these patches of wetland. We then compared that to controls which had no steel cable, and that allows the flamingos to enter and leave these patches of wetland easily. And they actually, we then check and observe, and it has been shown that flamingos are actually able to enter these control plots. So we compared these exposures to each other. Our hypotheses here were that within the exposures, when there actually is the steel cable that is keeping out the flamingos, we'll have higher invertebrate densities. And because invertebrate densities within the exposure are higher, we'll also see higher benthic, carbon dioxide, methane, and nitrous oxide fluxes inside those exposures. These study sites were not just in the Doñana natural space. We had about 11 different wetlands that we originally built exposures inside of. There were over 126 exposures in total. I think that the majority of time spent on this project was building and maintaining exposures. Our 11 wetlands are shown here. We were working in the Marismas del ODL, actually in the salt pans there in ODL. We also had exposures built in Lucia del Lobo and Caracoles, also at Laguna de Santa Olaya within the reserve, 3 ponds in Beta La Palma, and then also 2 in Bahia de Cádiz, in the Salinas in Bahia de Cádiz. And these sites were chosen because they represent a wide range of conditions. For example, just looking at salinity alone, in ODL, the salinity can be greater than 130 parts per thousand, even greater sometimes than 160 parts per thousand, which is about 4 times as high as the salinity in the ocean. Flamingos love these sites because they're full of chironomids, and that's one of the preferred food items for flamingos. Also, Beta La Palma has a salinity slightly just less than the ocean. The ocean is usually around 34 to 36 parts per thousand. Beta La Palma is just slightly lower and highly variable. We were working in 3 different the ponds at Beta La Palma. And then, of course, we also have our nearly fresh wetlands within the Doñana natural space. For example, here we're seeing Lucio del Lobo. And this is, of course, at a time when there is water. You don't often get to see it this way, it seems like. And this is one of the most challenging environments to work in because it's such a dynamic environment. Of course, it has water at certain times of year, but then as the summer approaches, it rapidly dries down. You can see some horse tracks just in the surface of the sediments here as it's drying. And then, of course, large parts of the year, and it seems like more and more, this is what Lucio del Lobo looks like. And I'm sorry, I realize that's misspelled on my map there. But you can see here that basically this is a dried out wetland, and these are some of my Erasmus interns, and I really relied heavily on Erasmus provided by 3SI, Erasmus interns provided by 3SI, and we were out here fixing exposures because when the wetland is dry, of course, horses and cattle pass through, and you cannot leave your exposures up or they get destroyed. As a result of this dryness that Spain has been experiencing more and more, we actually were not able to sample 3 of these wetlands in which we built exposures, although we spent about 2 years rebuilding them, setting them up every time we thought the water was going to come back, even though it never did. And so Lucio del Lobo and Caracoles were never actually sampled as a part of this study, unfortunately. All of this work would have been impossible without lots and lots of help. From other grad students and postdocs at the Doñana Biological Station. I mentioned the great, amazing interns I had from 3SI International, and of course, in the building of these and also the sampling of these mesocosms required, an army of people, and not just in the building, but also in the tearing down of all of these mesocosms, which I just completed this past summer. And you can see, of course, some familiar faces shown here. And especially I have to point out these 3 here because, Bia, MJ, and Cosme, I think we all nearly died of heat stroke several times, and they were there for most of the dismantling of all these mesocosms. And so I don't know how we still managed to have a lot of fun out in there in the field, but if you're listening, thank you very much, because I definitely would have died of heat stroke if it wasn't for your help. But we got it all removed mostly. And it was just overall a really fun project to be a part of. So I feel really fortunate to have been able to work at the Doñana Biological Station, and of course, all the help from all the directors, the staff, people who manage so much paperwork and everything, and field vehicles and everything. It's just a really fantastic place to work. So I feel very lucky to have been there, and I had fun. So what did we do exactly? We measured sediment to water, the benthic fluxes of the greenhouse gases moving from sediments into the water column, and also fluxes of gases moving from the water to the atmosphere. We did both of these things in this large manipulative experiment. We used this portable greenhouse gas analyzer in the upper left with a floating chamber to measure movement of gases from the water surface to the air. And we usually did this from a raft. You can see the edge of our raft here, the floating chamber. After we were finished measuring emissions from water to air, then we would jump into the water and we would grab some sediment cores, and we would take these sediment cores, which you can see in this photo here, back to one of the climatic chambers, one of the camera climaticas in the Doñana Biological Station. And we would then measure fluxes of methane, carbon dioxide, and nitrous oxide in these cores. So then we're getting the movement of gases from the sediment into the water. After we finished incubating the cores and measuring greenhouse gases, we then sampled the sediments for invertebrates. So we could remove all the invertebrates within the core and also analyze for carbon, nitrogen, and sulfur. And then in the water column, we also measured other things that might impact greenhouse gas emissions, such as chlorophyll, which is giving us an estimate of algal biomass, total and dissolved nitrogen and phosphorus, dissolved organic carbon, also dissolved oxygen, temperature, pH, and salinity. We basically combined all these things together to look at whether or not flamingos were having an effect, but also analyze what are the conditions that are contributing to greenhouse gas emissions in the Doñana wetlands. So this is looking at the effects of flamingos on invertebrate densities. And I'm going to show a couple different figures that are going to look similar here. This is looking at the difference between the exclosures, where we actually kept flamingos out, minus the values from the controls, where flamingos have access. So what you're seeing, if a value here on this plot, if a value is greater than 0, that means in the absence of flamingos, when the flamingos were kept out, there was an increase. So in this case, we're looking at the density of invertebrates per square meter, and in the absence of flamingos, those densities were significantly higher. We have all the bioturbators, and when I say bioturbating invertebrates, I'm talking about things that are actually burrowing into the sediments. We have chironomids, which are our aquatic fly larvae, particularly the large ones. We have aquatic worms over here, oligochaetes and polychaetes. And these were actually, we're analyzing the log transform data, which you can see up here. These densities of these invertebrates were all significantly higher when flamingos were kept out. And I should mention that this was about a 2-year exclusion for some of the longest lasting exposures that were in ODL. So this is a very long period of exposure. So yeah, invertebrates did significantly enhance the densities of benthic invertebrates as we expected. Now this is looking at the effects on greenhouse gases. So with the exclusion of flamingos, and again, we are basically looking at the exposure effect minus the control, and this is looking at milligrams of greenhouse gas per square meter per day. And we saw a significantly higher benthic fluxes, movement of gases from the sediment to the water column for carbon dioxide, and that's shown here in the center. And this was about 1 gram of CO2 per square meter per day. When flamingos were kept out, we saw 1 extra gram of CO2 per square meter per day. And that might not sound like a lot, but that's actually a fairly large amount of carbon, especially when you consider if you were to expand that over the course of a year. Of course, this is just looking at 1 extra gram per day. We could be talking about 300 extra grams of carbon. And then of course, express that across the entire extent of all the Doñana wetlands. And that could be a significantly higher amount of carbon moving from sediments into the water column across all those wetlands each year. For methane and nitrous oxide, we didn't see a significant effect of exclosure of flamingos. When we're looking now at fluxes on the right here, we're looking at movement of gases from water to the air. We saw emissions in the same direction, really not a great impact on methane, but we saw an enhancement of carbon dioxide. However, this difference in this case, although there were oftentimes higher emissions, we didn't see significantly higher. It wasn't statistically significant. There's a movement of gases from the water to the air in the absence of flamingos. Overall, looking at the different factors that affect CO2 flux, it was related to these different factors. This is the effect on benthic flux of CO2 in milligrams per square meter per day. And you can see that as water temperature is increasing, we see significantly greater emissions of CO2. With higher concentrations of nitrate and nitrite, we see reduced concentrations of CO2. With significantly greater densities in vertebrates, CO2 flux is enhanced, and with higher sediment nitrogen content, CO2 flux is also enhanced. And then we had this extra term, which is just in the model stating whether or not there is an exclosure, whether flamingos were excluded or not. And this extra term was actually retained in the model. I should mention that we are using Akaiki's information criterion to select among different linear mixed effects models. And this is going to penalize a model that is too complex. If a term does not significantly improve the performance of a model, it will be rejected. In our top models, this term that just said exclosure was always retained. And that suggests that there was some other factor that differed within the mesocosms, within these exclosures, that is not among these other things that we measured, something that has changed that is also contributing to this difference in CO2 emissions being higher when flamingos were kept out. That's because that this exclosure term suggests there's something that we didn't measure that also matters. But we explained 79% of the variability with the parameters shown on the screen here in CO2 emissions. For methane flux, we also have these four parameters explaining most of the variability across all these different coastal wetlands. Particularly salinity, as it increases, we see a sharp decline in methane emissions. With higher concentrations of nitrate and nitrite, we see higher methane emissions. Also, as the density of benthic invertebrates increases, we see significantly higher emissions of methane. And as sediment nitrogen content increases, we see significantly higher concentrations of methane as well. And so we are explaining about 94% of the variability in methane concentration across our study sites with these parameters. So in conclusion, going back to our original hypotheses, were invertebrate densities higher in exposures? Yes, this is particularly true for chironomids, oligochaetes, and polychaetes. Their densities were significantly higher when flamingos were excluded. Was greenhouse gas flux higher in exposures? Yes. Sediment carbon dioxide flux was significantly higher, and this is moving from the sediment to the water. But the flux of nitrous oxide and methane were largely, or at least not significantly affected by the exclusion of flamingos. But the water to air emissions in this case were not significantly affected. We saw changes in the same direction, but these were not statistically significant. So in conclusion, we saw this enhanced carbon dioxide flux in the absence of flamingos might be due to higher benthic invertebrate densities and invertebrate respiration because they're exhaling CO2 just like we do. But it's also possible, because we had that extra exclosure term in the model, it's possible that there's something different about the benthic biofilm community structure, like for example, the algae and the microbes on the bottom of the wetland that might be contributing to this effect. And that extra exclosure term in the CO2 flux model suggests that could be the case. We actually have collaborators at the University of Valencia and University of Granada who are analyzing microbial samples now to see if we've seen any significant changes in the benthic community of microbes. And that could happen because of that disturbance effect of the feeding activity of flamingos that I showed in my video earlier. Those microbial communities could be quite different because of that disturbance. And that disturbance, of course, would not be happening when the flamingos are excluded. Sediment, carbon, and nitrogen content were actually not significantly different inside and outside of the exposure. So I don't think it would be a case of a great difference in the total amount of microorganisms. It might just be a difference in the types of microorganisms that are present, or the actual structuring of the sediments or the microbial communities within those sediments. And also interestingly, greenhouse gas emissions were highest in the freshwater sites. And this was especially true during periods of low water, as you can see in Laguna de Santa Olaya. And of course, this is extremely important now in the face of climate change, because as you know, water quantity in the Doñana natural space is substantially reduced and is expected to continue to get reduced over time. And so this could be representing a significant and really important regime shift in the way that these ecosystems function. And just to talk a little bit more about that, basically all of the coastal wetland sites where I worked, we measured nutrient concentrations in the water, and all the sites where I worked, everywhere I sampled, was hyper-eutrophic. That means it's beyond what we consider a negatively impacted wetland. These are exceptionally high concentrations of nitrogen and phosphorus in these wetlands. And I was working often during times of low water. And one of the things that likely contributes to this is that we have more or less, the same density of animals relying on a smaller and smaller space in order to get water. So the cattle, the horses, of course, all the birds are being confined to smaller and smaller shrinking wetlands. And all of the guano, all of the excrement of the cattle and things are being concentrated into a smaller and smaller space. So the concentrations of nutrients as a result in those wetlands because of guanotrophication, and excretion are going to get higher and higher. And as a result, water quality is going to be impacted. And you can see this picture down here from Laguna de Santa Olaya. This is the greenest wetland I've ever worked in. During these times of low water, these nutrients are exceptionally concentrated. I think it's important then that, not just worrying about the quantity, and I know that many people are already thinking about this. These are ideas that were brought up, decades ago by other researchers at the Doñana Biological Station. It's really important to also think about the water quality that we are managing to protect the Doñana wetlands. The Flamingos project, that acronym, what that first M stood for was macrophytes, the plants, right? There were no plants in any of these wetlands. And I think and in the past, there were other studies, by Andy Green and colleagues, for example, in Veta La Palma, that showed that one of the effects of flamingos is reducing aquatic plant biomass. We were planning to test this. We thought we would see effects, but we never found plants in any of the wetlands we were working. And one of the things that might contribute to this is you all have heard from Martin Scheffer and other researchers and things that we have these alternate stable states in ecosystems. And with enough nutrient loading, if we're adding enough nutrients to the wetland, we see dominance by algae. And under these conditions, there's no light entering the wetland below the surface of the water anymore because it's being blocked by all these algae, and you can no longer have vascular plants living in the wetland. So we were never actually able to test the effects of plants on greenhouse gas emissions or the effects of flamingos on plants because we had no plants. And this could be a symptom of this nutrient loading and these changes in the Doñana wetlands. Also, these very short hydro periods or periods where there's water that doesn't allow the plants to survive. So with the short hydro periods and high nutrient loading, we might see increasingly the systems being dominated by a stable state in which algae are in control. And that might result then in reduced ability of the wetland to store carbon, because a lot of that carbon is actually stored in the vascular plants, not in the algae, and also enhanced greenhouse gas emissions. We saw there are highest methane emissions during times of low water. So very briefly, I don't want to spend a lot of time talking about this because I want to leave some time for questions, but what am I doing now in Kentucky? Well, I'm working currently to use satellites and remote sensing to try to determine from a distance the ability of a wetland to store carbon. And so we're actually specifically looking for what I just described. In this picture on the right, there are actually 4 wetlands in the image, but one in the upper right here is basically blending in with the grass because of all this fertilizer running off from the surrounding farm fields and from the surrounding lawns is enhancing algal growth. And we're trying to use these satellites then that fly over every 5 days and detect color to determine algal density and also as an indicator of methane emissions. Also working currently on projects where we have prescribed fire. We're burning sections of forests as a management tool, and I'm looking at the effects of fire on forest carbon budgets and methane flux from soils and also through trees. And comparing burned and unburned sections of forest, we burn about a 20 hectare section of forest every year, and then go and measure different effects. I don't want to spend too much time talking about that, but happy to talk more if anyone has questions about it. Also looking at we have a huge problem with the urban heat island, which I think you would all be familiar with, in Sevilla as well, effects on tree methane emissions. We're studying the effects on basically the urban core of the city of Louisville. It gets very hot during the summertime. And how does that affect the movement of greenhouse gases from soils and also as it passes through trees and other things in the environment? And then also working with some engineers here at the university on beaver wetlands and how basically beavers can help us better restore ecosystems and how the activity of beavers actually contributes to carbon storage within sites where we're trying to restore streams. And then finally, beginning to work a bit, and that's really going to be starting this summer, we're going to start testing our ability to measure emissions of CO2 and methane in urban ecosystems by mounting these open path sensors on the roofs of different buildings around the city of Louisville. So I have so many people to thank. Of course, Andy Green was my main supervisor at the Doñana Biological Station. And we had lots of different people who helped write the original grant. Emma Huertas at ICMAN, And also these two MVPs that I have to call out, Isa Reche and Iron Decline, because Isa from the University of Granada lent us a greenhouse gas analyzer, which is a very expensive piece of equipment. It's very generous of her to let us use that for extended periods of time. And also Iron at Wageningen University really lent a lot of equipment, portable greenhouse gas analyzer and other equipment that was vital to the project. Without them, never would have been able to get this work done. And of course, other people like Christoph has helped a lot with writing some really fancy code for quickly calculating fluxes from all the greenhouse gas data we collect. Jose Antonio, who's been analyzing invertebrate densities, Paco Hortas, and Marta Sanchez, who really orchestrated things at ODL and also in Cadiz. And then lots and lots of other people to thank. I've even left out many people here. And of course, the European Commission Horizon 2020 program. And I think with that, I'm happy to take any questions that anyone might have.
Okay, thank you very much, Andrew. So while the chat at the YouTube is warming up, I have a couple of questions. It really struck to me that a group of organisms, which I thought they would be our friends, are actually increasing the flux of methane. This is not organism, possibly unicellular algae. I thought that they were actually helping to fixate gases and not to fixate carbon, not to pass it to the air. How could you explain how this happens?
Yeah, that's a really great question. So algae are definitely going to help with pulling carbon dioxide out of the air because they are photosynthesizing, and when they do, they're actually removing carbon dioxide. The problem with algae is that when they die, they decompose very quickly. They're very easy to digest. They're very easy for microorganisms to break it down. And when you have really high densities of algae in the wetland, you have a constant movement of dying algae from the surface of the water down to the sediments. And that can then enhance the emissions of methane. So it's actually been shown in a lot of wetlands in some larger studies that the emissions of methane from lakes and wetlands is actually positively correlated to the amount of algae in the wetland. The thing is, if you have, for example, and you can see it in this picture, I'm not sure if you can see my slide, but when you have more vascular plants, like things like rushes, sedges, large plants, they're more difficult to break down. They decompose very slowly, so they tend to actually, a lot of the carbon dioxide that they remove from the atmosphere remains locked in the tissues of the plant, because you will still find those dead plants for years decomposing slowly on the bottom of the wetland, and that greenhouse gas then that would be released from their decomposition is not going to be returned to the atmosphere at nearly as high a rate. If that makes sense, it really has to do with how rapidly those populations of plants turn over. And the algae breaks down very, very quickly and then gets converted to methane.
Amazing. Another thing that I really enjoyed is that well, do you think that just aerating these large areas or solar ideators could just reduce enormously the amount of methane or something like that?
Yes, definitely.
In these places?
Yeah, absolutely.
Like that sounds like a very simple solution. I don't know if that's being used.
Yeah, people do use them in smaller, for example, in some fish farms, in some places where they raise fish, they will actually aerate the water. And that's done partially to keep the fish from dying, right? To keep oxygen in the water, but it also reduces the methane emissions. Yeah, if you could figure out a way to get some pumps out to these wetlands, you could do a large scale ecosystem study where we actually aerate the wetland and look at the changes. But yeah, it would work. I mean, it's a great point. You're absolutely right. If you kept it oxygenated, you wouldn't be able to produce as much methane.
That was amazing. And then just a last question before I get myself out of here, and leave other people to make questions too. That was another striking news to me. When you have these large animals compared to the microbials and algae and everything, storing carbon in their process, does this mean that the bigger the animal, the more proportion of carbon it's storing with respect to its respiration? Could we say something like that? Like as we have larger and larger organisms, they are storing more in proportion to what they are passing out to the air.
Yeah, good question. So that's getting into metabolic theory, I guess. We have to think back like some of the work by James Brown and others, and it's going to vary from one organism to another. But I need to look back again and think about exactly if it does scale with size. But I'm trying to think about the direction of the effect.
What I'm reading right now too is like I have been always thinking of, for example, cows as enemies to the climate change phenomenon, because they will be, producing a lot of methane, for example, or CO2. But if they are actually compensating this production of gases by storing a lot and passing it through the trophic chain to us, like,
yeah,
is this being accounted for?
No, I think you're right. They're generally bad. Yeah. So it's like, just like earlier when I talked about, our bodies store carbon, right, as humans, but that does not account for our impact on the environment. What we do by emitting, by basically digging up fossil fuels and burning them is far beyond the amount of carbon that our bodies can store. And basically all the farmland that it takes to raise cattle, all the fertilizer that we need to raise cattle, all of the emissions coming from cattle when they burp, when they belch out methane, that goes far beyond the amount of carbon being stored in their bodies. So no, they're not helping, unfortunately.
All right. But with respect to the amount, to the proportion of expelled versus retained, I see all these other factors that are actually absolutely negative for the environment and the climate. But I was thinking about the process of retention versus expulsion of gases, right? With the body size. I don't know.
Right. No, it's a really good question. And I think I honestly just don't want to tell you the wrong thing, but we can answer it very quickly by looking at the metabolic theory research. And again, James Brown is one of the people who pioneered this work, but we need to look at his papers, and it's a very quick explanation. I just don't want to tell you the wrong direction. As body size increases, that should directly scale to body size. But I can't remember if it is in which direction it goes. So I'll have to look that up again. Yeah. But a good question, and it does matter. So
I'm gonna leave other people to question too. I don't want to monopolize this. So Ivan Gomez Mestre comes with a couple of questions too. His first question is, how quickly would be the mat of worms rebuilt in each hydrological cycle? How would they recover in each hydrological cycle? I think that he means, his second question is there a massive die-off when the wetlands heats up prior to drying up? I think that he means he wants to know about what's the dynamics of the biomass of worms, cycling the nutrients.
Yeah, that's an excellent question. One of the things that we weren't sure about, for example, in that mesocosm study from the Netherlands, we didn't know if the effects that we were seeing were because of something the worms were doing when they were alive, or it is because the worms died. So we actually preserved all those sediments, and now I am counting through them to see what the worm mortality would have been. Because as these organisms die, they probably also are contributing to greenhouse gas emissions. And if they're dying in sediments, it might be this huge impact on wetlands. So Yvonne, I think that's a great question. I think that certainly during these times when the wetlands heat up and when they're drying down, there could be mass mortality events. These worm populations turn over very, very rapidly. And so you could see enhanced greenhouse gas emissions associated with dying. And I think that in itself is something that people don't often study. We often, if our study organism died, then the experiment is ruined. But I think actually understanding how greenhouse gas emissions are associated with death, I think could be a potentially interesting topic, especially in these highly disturbed ecosystems. So good question.
Cool. Thank you, Andrew. Ivan was also congratulating you for the fantastic talk. I also really enjoyed how you showed everything up very quickly and clearly. And Montserrat Villa, she has another question. She's also congratulating you, and her question is, how would the flux be if we add flux due to the flamingos metabolism? So she's a little bit with my doubt.
Right. Yeah. No, that's really interesting question. Obviously, I can't get at that with my chambers. The flamingos are also exhaling carbon dioxide just like we do. And I don't know. I don't know because they're also storing carbon within their bodies. And so I think that the flamingo body would be a carbon sink, right? They're actually storing carbon while they're alive. But the way to get at that would be to have a tower with open path sensors and actually measure the total emissions when numbers of flamingos change. But it would be so complicated to tease it all apart because there is the guano, which is a huge impact on these wetlands, and that's going to affect the whole ecosystem metabolism and also the disturbance effects.
You could measure it individually and then multiply by the number of flamingos, for example.
Yeah, potentially.
Have any individual measure and then multiply that. Since
we could.
The metabolic rate is gonna change not so much with temperature.
Yeah, and you might not even have to measure it directly because of, again, this I guess energetics and metabolic theory. We probably could just look at the body size of the flamingo and estimate the actual respiration rate and estimate the amount of CO2 that they're giving off. But yeah, excellent question. And getting at those types of things, it starts to become very complex. And I wish I could have done more of those types of things with this work, but we didn't quite get there yet. But it would be something really fun to pursue.
All right. We have another question from B. Almeida. She says, if I understood well, you said that in the exclosures, there were more carbon emission from the substrate to the water.
Mm-hmm.
But the emission from the water to the air is it as high?
No, it was lower. So the amount of carbon dioxide moving from the sediment to the water was higher than the amount of carbon dioxide moving from the water to the air. And there are a couple of different possible reasons for that. But I think in this case, you saw how green, the amount of algae in the wetland was very high. So a lot of the carbon dioxide that was released from the sediment, before it can actually escape to the atmosphere, it's probably being incorporated into algal cells. They're actually these photosynthetic phytoplankton and algae are taking up the carbon dioxide and they're sequestering it before it can escape from the wetland. So again, August, this gets back to what you were saying earlier about the algae sort of being beneficial. They are probably keeping some of that CO2 from escaping. And that would explain why the carbon dioxide emission from sediment to water is higher than carbon dioxide emission from water to air. But then a lot of it probably does eventually escape in some form as methane at some point. So
I see.
But yeah, it's a good question.
So exactly. Well, this answers the second question from B. Almeida, which was where is the carbon accumulating? Is it accumulating in the water? So it would be in the biofilm, in this biofilm, right? Or
yeah, probably in the biofilm or also in the algae in the water column. Yeah.
Okay. So Andy is coming with another question. He says that he was also adding to your answer to Ivan's questions. He said he was commenting that the change in the die-off or the change in biomass from the worms will largely depend on what kind of worms we are talking about. Like, for example, the chironomids can leave more sediments as flying adults, but the polychaetes should not or cannot. So yeah, apparently there is also changes, taxonomical or functional differences among the groups.
Yeah, no, that's a really good point. I mean, a lot of these aquatic invertebrates that actually can leave, like the fly larvae, they can detect the wetland drying and they might speed up their development and leave very quickly. So maybe the mortality in those groups might not be as high as with the worms or the crustaceans and things. But yeah, good point, Andy. Yeah, I agree.
That's it. Well, that's it then. Andrew, thank you very much. I think that you raised a fantastic conversation here, and I really thank you for your talk. So, do you want to give us any few words?
Just again, thank you so much. My Spanish is, as you all know, is not great, and the people at the Doñana Biological Station were always very helpful, very gracious, and very patient with me. And all the help I got over the years made this work possible. So I have hundreds of people to thank, and also don't take for granted what a great place the EBD actually is. You have some really fantastic resources and an army of field vehicles. In my biological department, we have one. And so, just having the ability to go out in the field, usually whenever you want to, and having these vehicles that can get you where you need to be is just an amazing thing. So I really enjoyed my time there, especially the great people. And so, yeah, I hope that you all, I know sometimes, field work is hard, science is hard, but hope you all feel lucky to be there because it was a really great place to be. So thank you again for allowing me to do this work. I really, really love the experience.
Thanks to you. People is thanking you back. And and I have to thank everyone for being here at one another week. And see you next week again, this time with Joan Porta, but also online. And hope to have you all around and have this fantastic conversation again. Thank you, everyone.
Thanks.